Okay, good evening, everyone. It should be live, so hopefully you can all see and hear us. So, evening, Craig. Evening, Sharon. Evening, Greg. Hey, Harry. Hey, Live the Real World. Hey, Steve. Hey, Susan. Sure. Anyone else? Hey, Mark and Victoria. Hey, Thomas. Hey, Kale. How are you all doing? Oh, hey, Michael. Oh, tonight we are joined by my good friend Steve Trim of Venomtech. It's actually second time on one of my streams, but yeah, I think last time was probably about a year or so ago. But
yeah,
time, Hey, Jeremy, and so hey, Jim. So, but invite Steve back on. So, for people who perhaps didn't see that stream, so do you want to just give yourself a quick introduction to yourself and what Venomtech is?
Yeah, sure. No, nice one. And yeah, it's great to be back. And thanks for the invite again. And so my name's Steve Trim. I'm the founder and chief scientific officer at Venomtech. And at Venomtech, we are a commercial venom supplier for principally drug discovery, but we also doing a lot. So over the last year or so, and a bit longer actually since we spoke, done a lot more in biopesticides and crop science and stuff, and that's quite a cool new area. So some cool new stuff to talk about, even for people who have seen the stream previously. And our cosmetic area is growing as well. So yeah, range of things, and we have all our venomous animals on site. So a collection of spiders, snakes, scorpions. We collect the venom from sort of milking like a herd, really recoverably, and then the science comes in where we separate the venom out into its individual component parts, and found how they are useful for the three market areas that we're working in. And something we've been doing since 2010. I spent 10 years at Pfizer as a drug discovery biologist prior to that point. So yeah, coming up into our 13th year now, which is quite exciting.
Yeah, that's quite a long time as well. So
yeah.
People or Harry just asked where Venomtech is based. We'll just say it's not open to the public. So
we're not a zoo, unfortunately. Yeah, we don't have a zoo license, so it's.
So, just in case anyone was thinking, hey Tron, thanks for that. I'll better show that up. Cheers for the donation, mate.
Oh, cool. So yeah, we are based in East Kent in the UK, and the old Pfizer site now called Discovery Park. So it's a scientific bioincubator space, really, I suppose. And yeah, as Phil rightly said, we're not open to the public because we would need a zoo license if we started doing that. And so our animal collection is purely a working animal collection for venom production and stuff. There's a few photos of the animals on our website. Not many because it scares quite a lot of our customers, interestingly enough. That's something we learned quite early on.
Fair enough. So, how did Venomtech come to be? How did you come up with the idea of doing this sort of thing?
Yeah, so back in my Pfizer days, my focus and experience is in pain and neuroscience, and looking for more sort of improved pain therapeutics was the key thing. Things with lower side effect burden. So opiates kill pain in a lot of people, but they also cause a lot of side effect problems, some of which prevent you from being able to work and actually function in normal life, as well as obviously dependency side of things. So and that's all what we call side effects. So it's due to the drug hitting more than one target. So there's a target that's killing the pain, and then there's a separate target that's causing the dependency and the dysphoria, and there's what we call central nervous system problems in your head. And then there's also other targets that are causing the gastrointestinal distress and chronic constipation. When we're talking about, it is severe. It's not, it's not to be a side effect to be taken lightly. So we're looking at drugs to sort of make the pain go away and not cause these other things. And it turns out from a quirk of evolution that spider venoms were quite useful in this area of actually hitting one pain point, not others. And the problem was getting hold of spider venoms in a research-ready way, basically, in a ready-to-use format. And so I pondered on this idea, and I thought about it while I was at Pfizer. We didn't really do too much in that line. We weren't definitely going to be bringing those sort of animals into the lab at Pfizer. And so it just sort of sat as an idea there, really. We worked with a few toxins, and I was a safety officer for the toxins that we did have in, so that was good training there as well. And at the same time, I'd been keeping non-dangerous spiders and snakes as pets. I still do, actually. And so when I got made redundant from Pfizer, I found myself with 3 key skills. I understood the biochemistry and biology of toxins and how they were useful in drug discovery. Importantly, I understood the safety of working with these things, and I understood I had about 10 years experience of exotic pet keeping at the same time. And so I put the 3 things together. So it is my professional life that drove the evolution of Venomtech, whereas my pet keeping was a skill that helped me do that, basically. And we set up in the back of a pet shop because that's where the animals were. And a good friend of ours said they had a bit of space at the back we could make use of, and set about working out safe working practices. And one of those actually led us to get a patent on a safe method of feeding venomous snakes. Because to me, coming from a non-venomous world, it was quite scary how people were keeping and feeding venomous animals. And in fact, some sort of well-known procedures and stuff in place, but it was like, from a safe working practices, you're supposed to separate yourself from the hazard, and opening the door to the mamba and offering it a rat, from my point of view, was not separating yourself from the hazard. So I thought of a better way of doing it. And it's just like how zookeepers feed other dangerous animals. They don't go in with a lump of meat on their back and sling it down for the tiger. The tiger is in a separate cage, separated away. So we devised a 2-way gate system that bolted onto the front of the snake enclosure, had a piece of koi plumbing tube that was big enough to put a dead rat in, and a screw cap on the end. So you could unscrew the cap, put the rat in, screw the cap up, and then open the slide gate, which then presented the rat to the inside, and the snake still being contained. So the first time we fed a mamba without opening its enclosure was a day to celebrate. And we actually got a patent granted for that. And we let it lapse because it's a statement of our safe working practices. So if anybody wants to pick up that technology, it's free for use. The information's out there, because it just made sense that if you don't have to open the door, you shouldn't on venomous animals like that. And what we also found is that because when we collect the animals for venom collection, it was different to the feeding, which now happened in this hole. You didn't get a strike response when you opened the main door. As soon as you rattled the feeding gate, that they were there and at it, with that sort of feeding response as you'd expect. But when you then open the lid or the door, they were quite different and sort of more quizzical, and it made better animals to handle as well. So so that was sort of a busy introduction to keeping the non-dangerous stuff, but the dangerous stuff, but the non-dangerous stuff is the bulk of what we actually keep. So a lot of theraphosid tarantula spiders are the workhorses of what we're doing. Because there's a big difference between them being venomous and dangerous, and having the ability to deliver venom into their prey makes them venomous, but it's only if that venom is toxic to humans at that dose is it actually a dangerous animal. And so therefore the bulk of what we do is actually working with the non-dangerous animals that a lot of your viewers will be keeping as pets, and I know you do as well. And so we just went out to Harm with an estate car, my estate car, and just filled it and drove back.
Just as you do.
To start the lab. And that was back in 2010. So That was hilarious coming back through customs, and they asked us where we've been, so we told them, have you got anything? Yeah, mostly spiders and snakes. Anything dangerous? Quite a lot of it. And they just went, on you go.
Yeah, we're not opening that box to see what was in there.
Pretty much that was the response. Yes, there was back in those days we were obviously in the EU, so there was no transit paperwork needed, so there was nothing wrong with what we were doing. It just seemed crazy.
Just like we've just got a whole box of venomous snakes in here.
When you open the boot, the rattlesnakes rattled at
you. Well, Jeremy's actually asked, we've kind of gone into this a little bit. What species are you currently working with? All kinds, whether scorpion, spiders, etc.
Yeah, so on our website, if you go into Venom products, there is a list, and I'm not going to recount them now because it's nearly 200 species.
Wow.
But it's, to put it into context, it's most of the mambas, most of the cobras, and nearly most of the rattlesnakes species, not just available species. And then we come into invertebrates, and we've got about 8% of the known theraphosid or tarantula species, because I think we're sitting at nearly 1,000 known species of theraphosid spiders. So I think we're sort of 7 or 8% of that. So it's still a big collection, but there's a lot more we could have. And then scorpions, we have about less than 20 species, I think, sort of less, a sort of smaller part of what we do. And then a handful of true spiders. We quite often have colonies of Heteropoda, Hogna, the wolf spiders.
Jeremy will be very interested in those because he's certainly working on breeding a lot of trues at the moment.
Oh, excellent! Yes, definitely give us a shout. Be successful with that.
His trues and working with Hogna and all of those sort of things. So he'll be his will have pricked up at that bit.
Yeah, cool, excellent stuff. And then we've also got permission from Natural England to work with British spiders.
Nice.
So we've got a nice colony of a couple of state-odor species, Eratogena and Arenaeus stuff. So venom collection from them is on the challenging end of things. So if you think about sort of a centimeter cube size, that's a milliliter. So from those spiders, we're collecting about 1,000th of that. So we would call that a microliter, and sometimes less. So sort of a 10,000th of a mil is the sort of volumes we're working at. So it's from Eratogena, the droplet of venom will sit on a pin tip, and it'll hang unaided. It's that small. It's quite complex skills, and there's not a training school. We've had to sort of essentially work this out. We've been doing it for a while. So.
Steve W. thought they used to live in Ramsgate.
Yeah, I used to live in Ramsgate too. I've moved.
So hey Tom, hey Catherine. More people. Just catching up with the chat, but Harry's asked, do you work with centipedes?
Yes, so we do, and they are definitely venomous. The more interesting, what is hilarious. So for animal welfare as well as staff safety, we anesthetize the animals or the invertebrates to collect their venom. And under anesthesia, centipedes are like jelly toys. They're just really floppy. They lose all structure. They're like the sort of rubber toys you get in the zoo gift shops. It's hilarious. They suddenly liven up and they come around the other side. Whereas the arachnids, having more structure to their exoskeleton, they're not too different under anesthesia. The centipedes are just really, really floppy. It's really bizarre. We've also done a little bit of work with millipedes, being a group of non-venomous animals that we've worked with. And you can stimulate those toxins to be produced, but it gets shot out. I think the best way of describing it is like a broadside fire from a pirate ship that it just shoots out sideways. which is quite challenging to collect, but we did work out some nice techniques to do it. And it's bright yellow, and anybody that's been stained by any of the giant millipedes will know the wonderful colours involved. And it's quite fascinating stuff. We don't know too much about it. So as far as we're aware, myself and my wife are the first people to realise that the millipedes had proteins in their secretions. So they're not just sequestering stuff from their plants. They must be making some of it themselves as well.
Oh, wow.
Unfortunately, that's so novel, we don't know what it is.
Well, that just makes it even more interesting.
Yeah, that's what we did back in 2012, I think.
Grace worked for a homeopathic doctor many years ago, and they used spider venom and snake venom in medicines for certain ailments.
That's an interesting point. And the dilution factor makes it sort of questionable how much venom's in that.
Steve put this for a reason, because I was actually me and Steve went on a little trip out yesterday, and we was even discussing this as a question that sometimes gets posed.
Yeah,
some people to see what the reaction is.
Yeah, it gets posed quite frequently, doesn't it? And it depends who's asking to how facetious I'm being. I think some of the Rhabdophis might be poisonous, aren't they?
Yeah, they are. Yeah.
I don't know too much about it, but this is not something we've worked with. But it is always an interesting thing, and there's some great cartoons about the difference between venom and poison.
And I suppose it's always seeing various reptile groups just to see who goes, there's no such thing as a poisonous snake, and then someone goes, what about Rhabdophis?
Yeah, exactly.
There's not many poisonous snakes, but there are some.
No, but on that line, you've also got poisonous birds as well, which would be quite cool.
And there's even there's venomous frogs as well, which is like.
Yeah, so the frogs that I think cask-headed frogs that headbutt venom into the prey.
Well, once I was at the Natural History Museum in London, and yes, we ended up with Steve Lane, and he was like, "Oh yeah, I've got a venomous frog." I'm like, "You what?" Headbutts. I'm like, "Oh my God."
That's awesome. I remember reading that paper. It was just incredible. That's the wonderful thing about evolution. There's no predisposed plan.
Told you he would, his ears would prick up.
Both.
He's breeding. He's working with both genus at the moment.
Yeah,
to get them breeding.
Yeah, I'd love some of the Australian holoconia as well. They get pretty massive. You don't see them in a hobby very often. Occasionally they do turn up, but there's the old one that turns up as a stray as well.
You may miss it, but how do you knock out centipedes in the hobby? You use CO2 or drowning, which I'm not a fan of drowning.
Wouldn't go with drowning.
There are people that can't come underwater to like knock them out. It's like, don't do that.
Yeah, so CO2 is recoverable in centipedes. You just need to keep an eye on dehydration because their exoskeleton's thinner than the arachnids. But yeah, so yeah, CO2 works fine as long as it's administered carefully.
Well, with something like not that hobbies could get it, but in the zoo world, like something like isoflurane, would that do a similar thing?
Yeah, so that's something we are looking into too, because so one of the other things I do is I'm chair of the Veterinary Invertebrate Society, and obviously there's a group of vets. They routinely use isoflurane, and as sevoflurane is a later generation organic anesthetic, and we still don't completely know how these work. Well, we know a bit more about how they work versus CO2.
Yeah, and we sort of discussed with Benjamin. Yes. We had Benjamin on again, sort of thing, and we sort of discussed how to anaesthetize and stuff, and what he sort of favours and stuff. So it's always interesting.
Yeah, it's the organic anaesthetics. We know from human use how they're working, and what they do is produce a situation where an animal, be it human, invertebrate, or otherwise, or anything in between, is immobile, unconscious, and unresponsive to pain and stimulus. And if you use that as a definition, then CO2 is being an anesthetic.
Yep.
But it's fully recoverable. But CO2 is definitely not an anesthetic in mammals. But if you use nitrogen instead of CO2, because assuming that it's just asphyxia, you get something slightly different. It's similar, but it's slightly different. So it's not just asphyxia. It is something else as well that we don't completely understand, and we're trying to work on what is going on in that particular situation. So it is, it is quite interesting, but it's nice and safe for the staff to use, and we've got carefully titrated protocols that we've used for years, and we've got spiders that we've worked with for the full 12 and a bit years we've been doing this, and they're still with us and still producing venom. So there's it definitely has a lot of characteristics of anesthetics, and we're still unsure of exactly what is going on, and that's partly because the invertebrate physiology is so weird compared to mammalian physiology. We don't really understand that either. So
That's true. Hey, Brian's asked about Komodo dragons, about whether they're venomous, poisonous, or toxic, considering the uniqueness of saliva.
Yeah, that's a great question. That'll lead into a great paper we had finally published last year. So I would say that the dragon is venomous. They have venom glands, but they're not poisonous, so they don't carry a poison. It is actively delivered. And the big debate that was going on, whether it's bacteria or venom, and the real answer is both. So from our own work, we've looked at venoms of cobras and spiders and found bacteria happily living in the venom. And this took about 6 years, sort of pilot project in the background, but finally got fully published last year, which was nice. So we've got the venom microbiome, so a microbial colonization of snakes and spiders, and we've potentially in scorpions as well. And as there is no common ancestor between venomous snakes and venomous spiders, it's probably in a lot of other venom systems as well. And we actually produced an agar plate containing venom and showed that the bacteria from the venom gland would grow on this, and the same species, but from a clinical isolate, wouldn't. So these bacteria have evolved to live in the venom gland, and that's probably why venoms have antimicrobial peptides, that the venomous animals are trying to defend against this bacterial infiltration. And so you end up with medically useful antimicrobial peptides in venoms because they're colonized by microbes. And I strongly suspect that the Komodo dragon situation is exactly the same, that they do have a venom system, and they do have bacteria that colonize that venom system. And so it is quite often evolution is not quite so clear cut as to what is actually going on. And it's obviously difficult to take samples of the prey animals to try and work out what is the actual cause of death and what's going on. It's a bit difficult to do a post-mortem with a dozen dragons around you, I would imagine.
Yeah, it's just a little bit. Yeah, it's something that certainly over the past few years kind of been argued over and over and over again, sort of whether varanids, just in general, the whole of varanids, whether they're venomous or not, kind of thing.
Yeah, and there's something with we did some Heloderma extractions a good few years ago now, and we were both sort of going in expecting that if we saw something sort of vaguely saliva-like, we'd call that a success. But we gave this thing a rubber tube to bite on, and it just poured out. I was like, wow. It sort of gave cobra level about 1 mil of venom out of this, an adult Heloderma. And they definitely earned the title of venomous. There was no doubt that was more than saliva. It just poured out.
Wow.
And then we pretty much left it with the rubber tube because it won't giving it up anytime soon.
Fair enough.
We no longer need it.
Hey, Jackie. Cal's asked, how was it discovered that spider venom was specific?
Now, that's a really good question, actually. And I've done a little bit of investigation into origins of venom research. So I've written a couple of book chapters on venom use in drug discovery. And it's obvious with snakes and the medically significant spiders and scorpions that the research was looking at why are they dangerous to humans, how is that working, and how can we stop it. Whereas understanding how spider venom is specific to pain receptors, which I guess is where your question's coming from based on what I was saying earlier, that's been actually through understanding and a desperation really to try and find things that are specific to these pain receptors, because classical drug discovery has struggled to solve that problem. And so it's actually through drug discovery screening where you actually set up an experiment looking at your receptor of interest, and then you look at the closely related ones that control heartbeat or brain signaling, for instance, and looking for things that hit one and not the other. And they sort of found out through that drug discovery mechanism. And that's the how, but the why is also a very interesting question. Sorry, a quick drink. And what I think is going on is that venom has evolved to either kill prey or deter predators. And it just so happens to be that those ion channels and targets in the prey have also evolved in humans and just through serendipity been involved in pain processing. So a venom from something like let's think about a Thrixopelma, or that's a really good one actually, that is reasonably innocuous to humans. The bite reports are quite mild, whereas it obviously quickly kills its invertebrate prey. But if you separate the venom out into its component parts, some of those component parts are exquisitely selective for a particular gene called NAV1.7. And this is a gene where they've been found in families that have a congenital insensitivity to pain. This is the gene that's the problem. So in other words, they are, they're fit and healthy people apart from the fact they're not feeling pain. And so it shows that this is a good target for drug discovery. And yeah, this venom is really, really active at this point. But there's no evolutionary reason for that to be the case. So the term is anthropocentric. So there's no human-based need for evolution to be beneficial to us. It's just not how evolution works. So there must be something else going on. And I think that the real answer is still out there to be found. But it's probably connected with the fact that receptors in the prey have also followed an independent evolutionary path and turned up as disease-relevant channels in humans, for instance.
That's really cool.
So that, yeah, it's really quite fascinating stuff of how that's happened. And yeah, with the antimicrobial thing, we've actually now, and it's taken us, say about 7 or 8 years to sort of working to find out why they've evolved antimicrobial peptides, but we always had a suspicion that it was something to benefit the animal itself. We didn't completely know that it was defending against microbes growing in the venom gland. And why they're pain relevant channels, it might be because of the evolution of targets from the prey. It might be something we've not thought of yet. It's definitely out there to be discovered. Whereas the pain causing venoms is a bit more obvious. So venoms like those from Psalmopius that sort of act like chili. I saw that in the comments.
Next one.
Yeah. It's very obvious to see that an injection of a chili-like compound is going to cause a predator pain and make them go, okay, this is not the easy meal I thought it was, and walk away. So it's actually, it could be more directly related to the human versions of those genes. And some great work by Wolfgang Worcester and a lot of colleagues about the evolution of spitting cobras or spitting in cobras is directly correlated with upright apes and hominid evolution. spraying venom at the eyes of a predator seems to be directly connected with us two-legged predators poking them with sticks, I guess.
Yeah, on the sort of chilies, so definitely our chili head, and he is asked when is the Venom hot sauce coming back?
Yeah, so it might do. We've not closed the door on it completely, but obviously, as we came into COVID, dealing directly with public sales and stuff was just not practical for obvious reasons, and so we sold up the stock we had and have not made it since. For us, it was always just an interesting marketing tool. Until it got picked up by Lad Bible and just went bonkers. And then it caused us more work than it was possibly worth in the long run. But it was fun, and as a marketing tool, it definitely did its thing, and a lot more people knew about Venomtech as a result of the chili sauce. I really like it. It was just good sauce as well.
I mean, I don't do spicy food, I can't do heat. I end up with a bottle because I was like, actually, you know what? It doesn't blow your head off, and it was really nice. But for anyone who doesn't know what we're actually talking about, how did you sort of come up with a Venom sort of chili sauce?
Yeah, it's a fascinating story. But so going back on the heat side of things, that was the only negative comments we ever got is it's not that hot, and it's like, well, we never said it was. We just made a nice, decent chili sauce. But people were there daring each other to try it and stuff like this, and it got a bit out of hand. And I did want to produce a follow up that would teach them a lesson, but I didn't quite get around to doing it. One day, that's on the list for future things to try. But it came about from me doing a lecture as part of the Institute of Directors, talking to non-scientists about what we do. And so I used and it happened to be in a food establishment. So I used the explanation of chili sauce is actually a neurotoxin. It's attacking a receptor in your nerves that detects thermal pain. So if you accidentally stick your hand on the stove, you'll pull it away because it's hot. And that is the same receptor that the chili binds to. It's called TRPV1. And therefore, the chili is actually causing you pain. It just so happens to be that a large proportion of the population like that pain for some weird reason that it doesn't make evolutionary sense. But I'm not a massive chili head, but I do like a lot of spicy food. And so I sort of started to talk about chili being a neurotoxin, and then saying that there are venoms that also hit the same receptor. And these have evolved, as I just mentioned, to cause pain in predators. So therefore, the predator would go away and not eat the spider, basically. And so at the end of my talk, I then sort of went on to talk about other venoms acting on other targets and sort of more about the sort of biochemistry of what we do. And at the end, the host came up to me and went, "We really need to make a venom chili sauce." And I was just like, "No, don't do that. That's a silly idea." For a start, the amount of venom you'd need, and getting it through testing and making it safe and all this sort of stuff. And basically, he wouldn't let it lie. And we came up with the idea that because the venom acts like chili, therefore chili also acts like venom. And so the branding became Steve's Scientific Sauce, based on the fact that it is acting like a venom does, but underneath it all, it was just a decent white label chili sauce. We tried a few local producers, we found one we liked, and then put the branding story behind it of how venom acts on TRPV1 and all this thing together. And that was that. We were making it in sort of 30 or 40 bottles at a time in a little farmhouse kitchen, as a bit of fun for conferences and stuff. And then I remember going to a conference, and and my phone was just ringing off the hook all the way. Of people, sort of Texans wanting pallets of the stuff. It's like, we're making 30 bottles at a time. We can't do pallets of the stuff. And yeah, when I got to the conference I was going to, it turned out that the website had been picked up by Lad Bible and just sort of completely blown beyond anything we'd prepared for. But it got me some good after dinner lectures. I delivered on the back of the chili sauce to send it at conferences and things. And yeah, it might be back at some point, but not in the immediate plans anyway.
That's cool. So John, who's in Norway, asked to work with Vipera.
Yes, we do. Not much. We do a little bit from here and there. We've got them, and we've collected the venom. But what drives the venom we select is the targets we're working on. So rather than the actual species, so it doesn't turn up very often in what we do, but it is there occasionally. Yes.
Sure. Harry's asked if you work with.
Oh yes.
I've heard of those. Which one's that one?
That's the Deathstalkers.
Oh, is that Deathstalker? I should have recognised that.
Yeah. Yeah. It's since got it was a monotypic genus, but it's later been reclassified in the sort of subspecies have become species and stuff. And I'm not completely sure where the taxonomy is at the moment, and that's why we employ a taxonomist to answer those sort of questions. And a great story
can be challenging at times.
Yes, when you find yourself counting scorpion anal arches to identify the species, you've come to a very specialist job. But one of the funny things with this was working with the BBC and a program called Pain, Pus and Poisons. Dr. Michael Mosley hosted it, and it came out probably 8 years ago now, I think, something along those sort of lines. And they wanted to show Illyrius walking over a CT scan of a patient with brain cancer, because chlorotoxin from the Deathstalker scorpions has been used with a fluorescent label because it binds to brain tumors and not normal brain tissue. So under infrared light, the surgeon can see the tumor fluorescing and can see that the normal tissue is not fluorescing, and therefore see where to guide the knife. Because in brain surgery, every extra piece of normal tissue you take is somebody's sort of essence of being. It could be speech areas, motor areas, thought processing, memories, all these sort of things. So you really want to take just the cancer. And so to illustrate this research, they wanted us to walk a Deathstalker over Michael Mosley's lap, and we thought, well, there's probably a safer way of doing that. They built a nice Perspex tall-sided box and put the CT scans underneath and let the scorpion walk over the top. And yeah, it keeps getting shown on BBC4, keeps turning up every couple of years, or get re-shown again. And the funny thing was spending about 40 minutes or so in a room with Michael Mosley, just going over Greencrest striatus. to get the pronunciation of the species, and it was good fun. He was really great to work with. So yeah, it does turn up from time to time. But yeah, they're great species to work with. But fairly innocuous to look at. They don't look, to the untrained eye, they don't look much different to a lot of other yellow scorpions. You've got to look really closely. So, yeah, they are iconic in their naming.
Yeah, definitely. A Scott, A Wolf, Hey Lewis. Oh, cheers, Lewis. Action up. Steve Doe's already got his first Cambridge. He's learnt about summer toxins that are able to help with strokes.
Yeah, nice. Yeah, they are still probably my favorite spider species. I have them as the screensaver on my phone. And they're just fantastic. They also have anti-malarials in their toxins as well. So yeah, and probably a lot of other stuff we've not found yet. So Yeah, fascinating species. I think they all look a little sort of gibbon-like in their sort of grey gangliness. Yeah, they're just great species. Quite agile too, which is also fun.
So Scott, I accidentally went straight past yours. Scott's asked any Venom research on into Alzheimer's or age reveals and brain age-related issues.
Yeah, so there is some work going on with amyotrophic lateral sclerosis, ALS, people doing the Ice Bucket Challenge for, as well as Huntington's. I don't specifically know about Alzheimer's, but definitely other neurodegenerative disorders. There are venoms that are showing some interest and some use. And the key thing there is that the venom peptides are quite large, and it's getting them into the brain and getting to the right side of action that is where a lot of research is going on. But the thing here is that the brain neurons talk to each other with ion channels, much like pain channels, and so therefore the venoms are quite useful in that area. And one of the venoms quite active in the brain or central nervous system, as we would call it, actually comes from the Nephilim, so the giant orb-weaving spiders. And that's really quite fascinating. And also, I love those species. I love seeing them in the wild. I've kept them a few times, and we've had them in the lab now and again. Obviously, they're quite a short-lived species, unfortunately. But their venom's quite fascinating, and it is that brain relevant component that helps them kill birds, basically, as well as the really strong webs. So yeah, it is definitely work in progress.
Oh, cool. I was saying if he finds a bottle of the sauce, he's going to drink it on live.
Well, if you do find a bottle, it'll be 2 years out of date.
See. Oh, Steve's he's just browsed your extensive venom library on the website. Do you keep all the species on the list?
Yes. That's why they're on the list.
Would you say it was about 200 species, did you say?
It's getting into that sort of region, yeah.
I don't know, he's not active in chat because feeding slings, where he's loving this and soaking up all this knowledge. It's incredible.
Yeah, cool. Yeah, that's
Harry's do you have any emperor scorpions? He's just recently got one.
Yeah. So we've got the Pandinus imperata, but also Viatoris, which have now been reclassified to Pandipalpus. My taxonomist tells me, which is really frustrating. And so this is one of the challenges we've had is that that our products change their names because somebody else decides. And that, as a business, is quite a hard thing to deal with. That we've got to keep a track of the taxonomy changing, because it means that the venom that we've sold from that animal is now called something else, and we need to keep a good database to be able to track it back through those changes, because over 12 years of doing stuff, some groups have been split and then put back together. Slightly differently.
Very recently, Heterometrus was completely revised, wasn't it, and stuff, and I'm no longer Heterometrus now and stuff.
Wolfgang Worcester at Bangor University has been doing a lot on revising the Naja species of cobras, and so it's great to get to a point where we've got the accurate name, but as we're going on that journey, we need to keep track of a shifting sand of name changes. But going back to the emperor scorpion question, their venom's been used for quite a long time in research. A toxin called imperotoxin, named after the species name of Pandinus imperata, actually acts on ion channels in muscle conduction, and these are actually inside the cell. So this venom gets inside the cell and acts on ion channels that control how the calcium signals within the cell. So it's really quite an important tool in research. So emperor scorpions have been used a lot, but imperotoxin is made recombinantly now by synthetically, so it's not we're not needing to milk tens of thousands of scorpions, which is good because that's pretty tedious.
Yeah, definitely. Steve asked, what protocols do you have in place to prevent escapes?
Lots. Yeah, so obviously I'm not a position we can go through in great detail, but just there's very simple practices that we always use secondary containment whenever we open a box. So if the animal gets out of the box it's in, it's contained in the secondary box, and simple things like that, along with checking and using remote husbandry tools and catcher devices that are always on hand. So yeah, it's a range of things from engineering controls such as using secondary containment through to staff policies and procedures at the sort of far end of things, because not only is it a business reputation problem if the animals are getting out, they are producing a product ultimately, so we look after them well because they are producing what the business, the raw materials that the business works on, so they are vital to our work, and we take this escape line seriously, even with live food, to make sure that literally nothing is out of the box it should be in.
That's really cool. Jeremy's asked, as he's feeding his slings, do you work with the red fang wandering spider?
Yeah, so yeah, because there are another ones that have their taxonomy has changed a little in recent years. Yeah, so all that sort of group of wandering spiders from the non-DWA through to Phenutria, we have worked with various areas, and that their venom again is quite interesting in the central nervous system stuff that we were talking about a few minutes ago, and fascinating species and quite good yielders. With regard to actual toxicity, we've not done any experiments to understand how toxic it is in whole animals, because we're really interested in what the component parts do in different ways. But yeah, they're a great species. I do like them.
That's cool. And you do work with the Phenutria as well currently?
Yeah. So the Phenutria we have worked with in the past, and they are the fastest animals I have ever worked with before or since. They move faster than your eyes can appear to resolve, and so therefore you need good controls in place. So we always anesthetize them in the home enclosures, for instance, so a sleepy spider can't escape because they're just so fast. It is bonkers, but fascinating in the same time. And I remember getting a Phenutria nigriventa from Andre Leitz many years ago. Poor guy, but he was saying, "Oh yeah, sorry, it's a bit small." And this thing was the biggest Phenutria I've ever seen. I'd love to have seen what he was actually intending to send. This was a beast, and still lightning fast.
Well, honestly, something that comes up all the time, and I'm sure people have it, in the chat, and anyone watching that's not in chat or anything, of Brazilian wandering spider Phenutria in bananas, sort of thing. For anyone who's wondering, how likely is that?
It is possible, but it's very, very unlikely if you think of the thousands of tons of bananas that come in. Someone like myself who's actively been looking for a free Phenutria.
I mean, for me, not necessarily. I once found a spider molt in a bag of bananas.
Where's the
spider?
I found no trace of any of them at all. But they are, the eggs that people, cases and stuff people see are very unlikely, A, to hatch, or B, to actually be what they think they are. But there is the odd occasion, and they are picked up Heathrow and other quarantine reception centres. That's what they're checking for. So nothing is perfect, and there is the occasional ones that have slipped in occasionally, but they're usually not in their best of health once they've been in cold chain shipped.
No, that's it. It's always in my job every now and then we'll get an email or a phone call being like, "There's a spider in our grapes or in a barn." It's like, yeah, it's probably native. It's probably a native one that's come in your house or whatever, or it's just in the market, sort of thing. So I was like, oh no, it's definitely a yellow sac spider. It's going to lay eggs inside us. Like, no, it's not. It's not. I'm not doing any of this.
Yeah, I love those great stories.
Constantly like, yeah, a really blurry photo. Can you ID this? No, I can't.
Yes, no, I only just tell it's a spider.
But happens all the time because it just goes everywhere through social media and social media just blowing it out of proportion.
Yeah, very much so. Yeah.
There's so many spider keepers, they're out actively sometimes looking for these things in produce. We don't find them ourselves.
So
it's been the most surprising discovery in Venom.
That's a really interesting question. There's lots that we can't explain, and yet the things that turn up the Venom does, and sort of to put it into context, that everything that we've tried to do with Venoms has worked in some way, shape, or form. So there's a lot more going on in Venom than we fully understand as a scientific community. But the surprising thing for me is quite complex biology, but it's about how genes are switched on and regulated, things called epigenetics. There was nothing in the literature about venoms acting in these pathways. So with collaborators at the Oxford SGC, as it was, we set about trying it, seeing what could happen. And yeah, sure enough, we could find venoms that could have a potential effect in what is quite a complex cancer pathway, which could be really useful. And it come from an area of not having any prior evidence at all. So yeah, that's really something that's quite surprising to me. But from a practical point, the most surprising thing was just how much venom came out of the heloderma. That was just like, wow. That was astonishing.
So this is going to Carol's asked, is it possible to build resilience to venom? I'm guessing that's come from some of the people that, especially in the States, kind of get bit, purposely get bitten.
Yeah. So the immune system's a double-edged sword. So the straight answer is yes, because this is how antivenoms are still currently produced. So you inject a horse or a sheep with a sublethal dose of venom. That causes an immune reaction. It's a foreign protein, and the animal generates antibodies to those venoms. We can then harvest those antibodies and use them to treat people who've been bitten. Now, if you are envenomated, a similar situation happens on the assumption you survive the envenomation event, obviously. And your immune system learns that this is something different and mounts a response to it. And the reason vaccines have to be very carefully engineered is you need to make sure the switches of the immune system go the right way. And so vaccines are carefully engineered to make sure the immune system goes into remembering that this is foreign and how to treat it in a measured response. The flip side is when you've heard people get allergic to bee venom, for instance, and then a sting from a bee can cause your immune system to recognize it as foreign, but in a much more dramatic way that causes anaphylaxis and shuts down your airways. There are reports of venom researchers getting anaphylaxis from venoms, particularly in countries where they don't use ventilated enclosures, so they're exposed to the dust of the venom. And so it is a double-edged sword. You're rolling the dice with your own health. That if you've had an envenomation event, whether it be deliberate or accidental, your next one might be better because the immune system has learnt to defend against it, or it might be a whole lot worse because it's going to flick into an anaphylaxis. And if you're doing an uncontrolled experiment, you won't know the answer until you test it a third time or a second time. So your immune system will always react to a venom because it's a foreign protein, just the same as it was if you injected somebody else's saliva. I wouldn't recommend that either, but the principle's the same. The principle's the same. You'll generate a foreign body reaction to those proteins. The next time you're exposed to those proteins, it might just innocuously destroy it as foreign, or you might swell up and die, anaphylaxis. So it is a thing left up to the experts with regards to vaccine immunity is what we're talking about, to make sure the immune system goes the right way and defends against the protein in future and not blows it out of proportion. But yeah, if you bear in mind people with anaphylaxis to bee venom, it's still possible with all the other venoms, then that'll keep you on the right course.
That's cool. Hey, Gallup. Okay, Harry's asked, do spiders dream?
That's a great question.
That is good. I like that, Harry.
And if you'd asked me before November, I wouldn't have an answer. But I have now. And the answer is probably yes. So this was presented at the Society for Neuroscience conference in November last year. And it turns out the bearded dragons also dream as well. And since learning this, I've watched my beardie quite many evenings. I've not seen any sign of it at all. And so when we're talking about dreaming, what we're talking about is REM sleep, or rapid eye movement sleep, because in humans, that's what we see. But if you put an EEG, so electroencephalogram, on and monitor your brain waves while your eyes are twitching in dreams, you'll see a very distinctive brainwave pattern. And so we can detect this pattern, we being the scientific community, can detect this pattern in many other animals. And people keeping cats and dogs will be quite familiar with their cats and dogs sort of twitching a little bit while they're sleeping. In this paper, this research group discovered, I can't remember the species they worked on. I think it was a true spider, and I can't remember what it was. But they actually went through phases of dreaming where their legs curled up in a death pose, only for a few seconds, and then opened out again. So they were showing those sort of twitchy-like movements, but actually it was sometimes a full death curl for a couple of seconds, and then neck out. But the electrical activity had analogies to vertebrate dreaming and REM sleep. So the understanding at the moment is yes, and if that turns out to definitely be the case, now we know very little about invertebrate central nervous system processing, then it probably opens up the fact that all animals dream. It's quite possible. And yeah, that's REM sleep is really important for how memories are segmented and formed and shuffled. A bit like clearing out the cache in your laptop. You can't store everything you see, hear, and do. And so your brain decides every night what is useful information, those things that get used more often, and what isn't. And what you'll find over time is that certain things you struggle to remember because your brain is deciding that it's actually not useful. Yeah, it's part of the dreaming process. But yeah, spiders dream, so possibly other arachnids may do too. But yeah, I keep looking out to see if my beardies or any of the snakes are sort of twitching or showing any signs of dreaming. It's harder with snakes
to actually see though, like especially with like a spider suddenly going in, just in there and suddenly going into a death goal and then out again, probably give any spider keeper a heart attack a bit.
Yeah. And I guess that if people were witnessing this frequently, that people would be posting about it because it would be so bizarre. But the movements in dreaming is a failure because our brains and all animals really have a molecular switch that stops you acting out your dreams. Because over evolution, this is a good survival thing. Because if you're sleeping safely in a cave and you dream you've gone out running, you're going to either walk into the walls, a cave water system, or out into the where the lions and hyenas and stuff are wandering. So there's probably a strong selection pressure to not act out your dreams because they're different environments what you're physically in. And obviously people who sleepwalk are on the far end of that, where the gate has failed and they are acting out a dream sequence. But what's a lot more common in animals and humans is that just sort of slight odd signals get through, just sort of little twitches going on. And so it might be just so subtle that people haven't noticed it. And also, you've got to watch an animal for a very long time to see if it's dreaming.
Yeah, and it's probably quite difficult as well, because you've got to have probably extremely, you're not disturbed the spider either, and so some of that sort of thing.
Yeah, because also the thing in this situation is that you need to be recording the neural signals to know that it's asleep. Because again, not having eyelids, how do you spell?
How do you know?
A resting spider from a sleeping spider.
Yeah,
well, I don't know the answer to that.
That's really, really interesting. What's been the most surprising animal to have venom? Unlikely candidate to have venom.
Probably slow lorises.
I've
known about that for many years after meeting Alan Akaris and seeing her present at Venomtech many years ago. And that's probably going to be one of the more surprising things. But then in later
work as well.
We
have slow loris here.
Yeah, I thought you did. But then in later years, Ron Jenner from NHM brought another surprise with the Glycera bloodworms. That was okay. Yeah, so that was like, wow, bloodworms are venomous. And under the microscope, they've got 4 hooks that are connected back to venom glands. So
they say bloodworms that people feed to their fish.
Yes.
Oh wow! Well, there you go. If you've got fish, you're feeding them a venomous animal.
Yeah. So there's lovely surprises like that. But I think some of the bigger surprises that came a good few years ago to me were things like in catfish. The venom has evolved 30 different times just in catfish.
Oh wow!
There isn't one venomous catfish predecessor as far as we currently understand. Venom is so powerful at protecting animals that it's turned up about 30 different times, which is just absolutely incredible. But probably the coolest thing, and I still think it's debated at the moment, and that is the Port Jackson cat sharks, Heterodontus portus jacksoni. And they've got a spur just in front of their dorsal fin, and there are, I think the research is sort of probably conflicting is the best description, but there's a potential to have venomous sharks, which is pretty awesome. It's not quite sharks with laser beams, but it's getting there.
It's getting there. So, well, that's always a nice hot topic for debate.
Yes.
As poisonous.
Yes. It may be pedantic, but because venom explains, it's the right word for the right thing, but also it explains more about the biology. When somebody describes something as venomous, that it has a delivery mechanism with it. And the confusing part is that some venoms are poisonous, but that's only when they're out of the animal. Yes, very much so. But it depends who's asking the question to how I phrase the response, but I think they should know better.
That's it. And I will point out as well, if you're referring to snakes, there are some poisonous snakes out there. They're also venomous as well, but
you've got to
kill back snakes. So Red Ophis are known to be poisonous.
Yes,
because normally the main one is people talking about poisonous snakes, and often they mean venomous.
Yes. Yeah. So when people ask me, do you have any poisonous snakes? I often go, no.
Question, what's your opinion on the mRNA vaccine?
Okay, yeah, I'm happy to take that. So it's a different psychic. So I think it's a fantastic invention of human ingenuity and medicine, and it is understanding how that immune system works is the key to this, and understanding that the immune system could go one in two ways, and if you present it in a way that's going to go down the defense route, it's a very powerful thing, and it's got us back to conferences and gigs and getting back out into the world. So yeah, it's a fascinating piece of really cool science when it comes down to it, and it's not just a COVID thing. It will, the like with so many sort of inventions, that the forerunning science was happening for a few years beforehand, in the background, people working out how these things work, can it be done, and to work it out. And so when when a massive need turned up, there was an opportunity ready to harvest, which is how we managed to do it, partly how we managed to do it so quickly, again, we being the scientific community, and the other thing is just sheer brute force. People threw more money and more people over more hours at something. Pfizer were running 3 shifts a day, so obviously you can do stuff 3 times faster without then putting more people on it as well. So yeah, I'm, my background is genetics originally, and it's just it's a piece of really cool science that's helped us get on with our lives.
Yeah, definitely, and that's it. With things like that, sometimes people think, "Oh, how did it come with this so quickly?" and forget or don't realize there's potentially 10 years plus worth of work before that. Actually, that's gone into it already and stuff.
Yeah, because they were looking at it for influenza.
Yeah,
because influenza changes so rapidly. You need a vaccine to be able. You need to be able to change the vaccine. So there's a new flu vaccine every year, based on the strains that are likely to be around, and this is a tool that would allow you to adapt a vaccine quickly. And so yeah, the background preliminary research was already starting to happen, but was not making headlines anyway, so people didn't know about it.
Yeah, that's it. I don't ask what that her husband has snake venom to stop his bleeding condition.
Excellent. Yeah, so there's a handful of drugs out on the market that are derived from venoms. My favorite being captopril, which is a high blood pressure medication, or antihypertensive to give it its proper term. And this is the first ever medication to cure high blood pressure, but it's also a venom derived compound. And there are a collection of either pro-clotting or clot-busting drugs from vipers, Echis being one of the Echistatin being one of the key ones, and Agrestat being another one. So yes, there are drugs from venoms out on the market. There's a type 2 diabetes drug from Heloderma, and a painkiller from a venomous cone snail.
As you're going on to that, I was asked about cone snails. As you said that, I was like, ah.
Perfect. Yes. So cone snails are amazing. Their venom is so dangerous to humans. It seems a little bit unreal, but evolution is not wasteful, and the reason these venoms are so potent and so powerful is that these plucky little squidgy snails have decided that they can take on bony fish. And the only way a squidgy snail can tackle a bony fish is if it kills it very, very quickly, which then gives you a very potent venom. We don't work with cone snail venom. I have done in the past, but they are on Schedule 5 of the Counterterrorism Act. So there's some quite serious legislation, and the Home Office get quite interested when you start working with cone snails. So it's not something we do in the
UK. I was even fan to Spider's Dream. Imagine the spider matrix while dreaming about chasing the cockroach.
Yeah, possible. But if you think about the sort of crazy bonkers things humans dream, the spiders could be dreaming about absolutely anything.
Yeah, that's it. Oh, hey Amy. Oh, I hope Matt gets better. See things. I'll catch up with the chat. I've got apologies. It's already been said, but has any work been done with Scolopendra?
Yes, so yeah, we did mention that earlier. There's some interesting pain-causing venoms from Scolopendra, which anybody who's been bitten by them won't be surprised at. But there's also antimicrobials and several other interesting compounds in their venoms, but they are much less studied than the arachnids at this current stage. But yeah, we've got several species, also Ethnostigmus as well. We work with a few different species. I guess you'd be able to watch back the chat.
Scott's asking, end the legend of the daddy longlegs being so venomous. It really depends on what you mean by daddy longlegs.
Yes, yeah, because I grew up with them being crane flies, which are not venomous.
They're flies.
That's it. So, deal with it.
Got some people refer to harvestmen as daddy long legs.
Yeah, that is true.
And then stellar spiders as daddy long legs as well and stuff. And
yeah,
daddy longs is the most venomous spider in the world, but it can't bite a human.
Yeah, they Phocus phalangeloides, aren't they? The stellar spiders. Yeah. So they are definitely venomous, and they can't really pierce human skin. But in terms of their venom, we've not really looked. But if you look, and one of the things that is quite fascinating with biology, particularly with native species, you can just go and watch and learn in their natural habitat. The phalangeloides will tackle quite big spiders. I've seen them win battles against bigger Atagina. But the trick is understanding and watching the battle. The phalangeloides are not biting the prey and running away, which would be the mark of a very powerful venom. What they're doing is that their massive leg span, they're just reaching around them and bundling them up. So they are biting them later, but they are overcoming these big spiders, which Eratogena are quite heavily bodied compared to phalangeloides, but they're just reaching around them and binding them up. So I don't think the venom is that potent. And also, we've not gone that small in our venom collection, because if you look at the fangs of phalangeloides, they are really tiny.
That's it. So yeah, he's put harvestman. So I'm guessing he means harvestman then, as
rather than phalangeloides. So harvestmen are not venomous. So if you look under the microscope, what's really cool is their Telisera have little claws on them, but they're sort of tucked up. And when you look under the microscope, you realize that they've got double segments like all arachnids have. Absolutely fascinating creatures under a microscope, but not venomous.
I'm guessing you don't really work with solifugids.
No, because they're not venomous either.
Yeah.
I'd like to work with as we're talking about other arachnid taxa, I'd love to work with pseudoscorpions. But again, the extraction technique is specialized, and that's coming from us.
They're tiny.
Because some of them can ride on the back of bees. They're really really tiny. But there's a wonderful photograph came out a few years ago, and actually I've used it in textbook. Of a device called a meso lens, MESO lens, and allowed the researchers to actually look through the exoskeleton of the pseudoscorpions and actually see the organs within, and they can actually see the venom gland in the finger of the mobile finger of the pectine. So it's like a scorpion having venom at the end of its pincers, which again that comes back to surprising venomous animals. That was quite surprising that their venom is in a completely unique place. And there's only a couple of labs that have actually looked at pseudoscorpion venoms, and mostly at the gene expression level, because you're not going to be getting much venom from an animal that's only a grain of rice size in the first place, or less.
Yeah, definitely not. Yeah, definitely not a lot. But how do you go about sort of extracting things like spider or scorpion venom?
So the spiders, it would depend on the taxonomic group. So Theraphotidae, the tarantula spiders, their fangs are parallel facing down. So when they're anesthetized, we can swing their fangs forward, put them in a vial, and use a very tiny electric stimulation. And what that does is just squeeze the venom gland. So it's basically like squeezing a balloon of water, and it squeezes the venom out into the tube. Araneomorph spiders, on the other hand, are quite different because they've got opposing fangs. And so we can't just put the pair in a tube. So then we actually have special attachments on each fang and do each side one at a time, but all under CO2 anesthesia. And the smaller the spider is, the better the microscope you need to to look at it. And then you get false hope, because you see this amazing venom yield, and then you take it out from under the microscope and realize that it was quite far zoomed in.
Oh, that's all it is. I'm guessing with like the snakes, it's the same as you see on TV, or perhaps.
So the key difference from us is that we have we're starting to see more and more YouTube videos with the venom collection device clamped to the desk, which is good, because some of these snakes just overreach the collection vessel, and if you've got the vessel in one hand and the snake in the other, the chance of them overreaching in the back of your hand is too high. So right from the beginning, we've had the pot clamped to the bench, and you milk the snake away from you. And they'll put venom into anything you point them at. The trick is holding the snake
safely,
in a way that you can do it again afterwards.
Yes. I was going to ask, what other hobbies does a venom expert have?
That's a great question. I do a lot of cooking, so particularly in lockdown, we sort of did pseudo traveling. So I learned to do Japanese cuisine, and got a few other things in like that. But my main hobby is playing drums. It's just something I share with Phil.
We have a lot in common. Like drums and metal music.
Yeah. Listening to metal.
So trying to ask thoughts on
Irukandji.
Yeah.
So Irukandji are tiny box jellyfish in the South Pacific. And the first time I read anything about them, I was on a flight to Bali, and saw about these tiny box jellyfish. And I do a bit of snorkeling as well, mostly in warm waters. But and after seeing about these sort of deadly tiny thumb-sized jellyfish, when I was snorkeling, my focus distance was about here, checking every little speck that went past me. But I never saw any then. But many years later, we went to Perth, and we saw several in Perth Harbour. So they are incredible. And again, much like the cone snails, you've got a small squidgy animal that likes to prey on other animals, and the only way it can do it is by having a very powerful venom that kills them quickly. So Irukandji deserve quite great respect, and because they have many meters of tentacles behind this tiny little bell on the top. And we've done some work. We've got a paper actually on recoverable methods of collecting venom from Cnidaria, so the group of jellyfish and anemones and corals. But they are very challenging species to work with, and definitely more in this space in future years.
So, hey Kitty, Greg's asked a good one. If you have so many spiders, how do you keep track of which ones have been milked and when? And also, what do you do with spiders that mature? Do you breed?
So it's quite simple. We have a database and we record data. So every animal has a unique ID code when it comes into the lab, and that stays with that animal for its entire lifespan, independent of if the species gets taxonomically changed. That animal will remain with that unique code. And yeah, whenever we've fed it, or we've done any welfare checks or collect venom from it, it's recorded in a database, and we know exactly when every animal has had something done. So we know not to collect venom too close after it's been fed, for instance, and what the when the last anesthesia was, when the last milking was, and it's just good, data handling, basically. Fairly, straightforward stuff. We've been doing it for 12, 13 years, but it was just really important right from the beginning, because venom takes time to reproduce. We needed to know which animal had been milked, and we've had many spiders that mature over the time, and as a result, most of our collection of Theraphosid is biased to females, because the males have grown old and died by now. We have bred some, but we don't do it often. As one of the viewers was saying earlier, that they weren't paying full attention because they were feeding spiderlings. We'd end up just feeding spiderlings if we were breeding everything we were keeping. And the wonders of the long-lived Theraphosid species is there's just no need. We have between 4 and 6 animals of each species, and they'll be fine for 20 years. Whereas the Aranimorphs, the true spiders, we do need to breed colonies because they live sort of 12 to 18 months. So there's a different approach for Aranimorphs versus Theraphosid.
Cool.
And also, if we hatch out babies, then they take a long time before they're big enough to actually use for venom collection as well. So they'd be taking up a lot of husbandry time, and we wouldn't get any use back out of them. So another reason for doing it.
A lot of that goes into it. So he's got to speed up on the venomous bloodworms.
Yeah, that's a question you're going to have to ask Ron Jenner about. I think he may mention it in his book Venom. Give a free plug to Ron. I'll get a beer off him at some point. But the honest answer is I can't remember.
I'm pretty sure I've got that book, so I have to have another look in that, because
that might. Yeah, cheers.
Yeah, Mark is a very, very big Iron Maiden fan.
Cool. Got mouse mat as well.
Jackie's asking about the blue-ringed octopus.
Yes, but not what you think. If you thought anything, let's go back. So the venom of the blue-ringed octopus shares a lot in common with that of the pufferfish, in that its principal component is tetrodotoxin, and that is used a lot in pain research because it blocks a certain subset of pain-relevant ion channels. The problem with tetrodotoxin, the reason we know about it, is it also stops cardiac or heart signaling ion channels, which is, if you have poorly prepared fugu sushi, it will stop you dead because there is no antivenom, and it just stops your heart from beating. Just straightforward molecular, shuts the switch off. And this is another venom, or in the pufferfish it's a poison, and in the octopus it's a venom, that is actually from a bacteria. Tetrodotoxin is produced by a bacteria that's found in a lot of marine sediments, and the pufferfish and the octopus have evolved to sequester this toxin and use it for their own means. But it's actually a bacteria that is producing this venom. So yeah, it is used for quite a lot of research, but not specifically as people think of tetrodotoxin coming from pufferfish. And an interesting thing that I only learnt whilst writing a recent book chapter on ion channels is that it should be called tetraodotoxin. The A has been missed out over years because the genera of pufferfish is or family of pufferfish is Tetraodontidae, which is what the toxin was named from, but it's been used so often that it's ended up with a slight short form, a misspelling almost, of tetrodotoxin.
Yeah, does it take long to replenish its venom, and does it have its own use-by date? So you can refresh it.
There's two parts to that question. So replenishing the venom from when it's used takes about 2 weeks, roughly. But it doesn't have a internal use by date in terms of the snake doesn't flush out a batch of venom and produce a new batch. The proteins in it will degrade slowly over time and be slowly replaced. So it's just sort of a tick over replenishment or refresh rate. So, for instance, during periods of anorexia, so non-feeding, the snake venom will replace some of the proteins, but just slowly, piece by piece. Whereas when all the venom is used in a envenomation event, whether that be manual collection or actually into prey or predator, then it takes about 2 weeks to fully replenish it. But the important thing is that a snake is never safe. They, even under human conditions where we're actually extracting the venom, they never release all of it. There is always something left in the tank, just in case.
Hey, Matt. Do you test the genetics of a venomous animal to make sure there's no hybridization before you start working with the individual?
So we've done a little bit on this, and we've worked up some primers and genetic tools on Poecilotheria spiders a few years ago, and a poster on this. But our main defense against this is actually understanding the lineage of the animals and keeping them on site. So if there is a hybridization event in there, we're not producing antivenom, so we don't need to mimic the wild state of the venom. We need to know what we found in that particular experiment, and we have the animals on site to go back to. So not directly, but as a geneticist, I would love to actually do more, get some whole genomes and understand more about the venomous animals, but it's just not required. So it's not and it's also very expensive stuff to do. So.
Yeah, but obviously, within the tarantula world, things like the curly head got the hybridized one, the hobby form, and see some people wonder whether some of the pokies have been hybridized or not, and stuff, and various other
and with brachypelmas as well as challenges there. And part of the problem here is evolution is a continuum, and humans like to put stuff in boxes. So we've looked at the range of Brachypelma vagans, for instance, and gone, this is the vagans and this is not. This is something else. Whereas evolutionary, they might be part of we're only looking at a snapshot in time. Give it another couple of million years, they might have fully formed into separate species or that. And so that's part of the problem. So our human definition of a species is a sort of an isolated unit that theoretically cannot produce fertile offspring from hybridization. However, in nature, some of those speciation events that they don't interbreed because of geographical boundaries such as mountain ranges or lakes, rivers, and oceans. And so in captivity, they might be able to hybridize. So it is a difficult challenge, and it's always we need to bear in mind that we're not looking at the end of evolution. It's just the current snapshot we're sitting in, and trying to make sense of what's going on. I would love to again go back to my genetic roots and have whole genomes for all the species we're working with, and we'd understand a lot more about which is which species and which are just color morphs of others. But it's a lot of time and money to do that.
That's brilliant. Hey, Pimble. He wants to say that this is an awesome stream. Cheers, Leah.
Thanks.
Cheers. Scott says very interesting stuff. Fantastic. So as you said, yeah, you work with a lot of paraphosids. So I'm guessing most of those are very common sort of hobby staple ones that probably most people in the chat actually keep in their collections.
Yeah, very much so. And we also like the dull brown jobs. And there's a good reason for that, because taxonomically they're interesting, and we don't need them to look good. We're interested in their venom. So, we have some wonderful pokies, and some of the red Tapinochenis, and some brachypalmas, obviously with wonderful colours. But a large bulk of the collection are shades of brown and grey, because we're not keeping them for showy species. They are for their venom interest. And yeah, a lot of them are very common in captivity. So that also makes it useful for us, and also interesting when we're talking about venom from Psalmopius, for instance, or Grammus dollars, and that people are very familiar with. The Grammus dolorosia, there's a component in that venom that actually is useful in cardiac arrhythmias, or sort of heart manifestations, because it blocks stretch-activated potassium channels. Which what this means is that when the heart beats, it stretches, and this causes a firing, and so sometimes it can cause it to beat out of sync. And if you look at this one component of the venom, it can actually start to bring that back into rhythm. But it's something you'd never notice on a bite. It's just not produced in anywhere near high enough amounts or anything. But when you separate out its molecular component, it's a really interesting piece of biology from what was a very common spider.
Less so. Certainly used to be a lot more common than what it is now.
Yes.
Like, remember the days where buy an adult for like a tenner.
Nowadays, yeah, very much so.
Sort of thing. I've got a couple, but that was actually quite interesting that recently. Was it Botarium rosea got revised all into rosea?
Yes. And that's not the first time that's happened.
No, this has happened so many times. So I was like, I think I might as well go Team Calcodes. He loves his Calcodes.
Yeah, no, there's the interesting thing with their venom as well, that there's a trying to think. There was it was sold as a subspecies called Paysoni, that was then sort of realized that it was just Calcodes. But the venom is different between Paysoni, and Calcodes. So from my point of view, I think the jury's out whether they are the same or different.
Yeah, but would that be similar? Because I know like things like Russell's vipers, depending on what part of India they're from, they can have like different venoms, can't they?
Yeah, very much
so. Sort of thing.
Yeah, so you do get geographical differences, definitely, and it turns up in a lot of species in the Bothrops, the fertile ant snakes, because their range is huge over a large part of South America, and sort of coastal forms and mountain forms have different venoms because they're living in different niches, and the error there is whether we're calling them a species or subspecies goes on the information we have at the time, and that might not be what's actually going on in evolution. We just don't have all the answers yet. The Russell's vipers are fascinating. I'd love to see them in the wild. Hopefully one day.
Yeah, they really, really are. So seeing some of the research done on them, we've had they can be completely different venoms to each other depending on what part of India, and they can even some can even I think like some insects can, depending on what they're eating, like over time can change their venom to match what they're mainly hunting.
Yeah.
So, it's really difficult then for the anti-venom producers.
Yeah, but so the anti-venom has to be particular to that local area then, which makes it more expensive because you have to make different batches for different regions. And we've got good collaborators at Reading University, and through that collaboration we've done some research on some interesting case studies of weird pathologies from Russell's viper bites in India, causing salivary calculus things that have to be surgically removed, and priapisms, as well as obviously the coagulation issues and stuff that Russell's vipers do. So yeah, they're fascinating snakes.
Greg just put, he's just put, but he also just put, lol, Phil answered about geographical differences.
Yeah, I'm not sure if pollution would be affecting venom, because the thing driving venom is its ability to kill prey and defend against predators, and that's what's driving these things. So if a snake finds itself in a region with very few mammals and a lot of reptiles and amphibians, then there's a selection pressure to drift the venom to being more potent to reptiles for that region. And you see with Echis, there's some quite a lot of research on Echis venoms, that the youngsters have a lot of arachnid and insect toxins, arthropod toxins, and then they only start expressing mammalian toxins when they mature and get big enough to tackle mammalian food. It's called ontogenetic shift, where the youngsters have got a very different venom profile to the adults.
Cool. So Steve's come in with one of his jokes about what species of tarantula could cure a headache, because he's got one on He's hoping he's got a tarantula that could.
Well, a bite from a Psalmopius or Pokey would definitely take your mind off it. Give you something else to think about.
yeah, definitely a Pokey would. Yeah, be thinking about that pain a lot. Yeah, have you worked with Sicarius?
So we've done some interesting work with Sicarius. We treated them like DWA spiders because we had Loxosceles at the same time, and their venom is very, very similar. They do the sort of Scooby-Doo thing when they try to climb a wall, whereas Loxosceles are quite adept at climbing walls. But other than that, their venom is very similar.
Well, going on, obviously, there's always sort of an argument about Sicarius, whether they should or shouldn't be DWA, and just how potent their venom is and stuff. And there's always a lot of very mixed information that people just in general hobby can get access to and stuff. How are they? Do they have a dangerous venom?
Yeah, so our approach was to actually, treat them as dangerous animals. They were locked in secondary containment, 2 members of staff whenever those enclosures were opened. The full works just exactly as we treated our Loxosceles, because when you look at the biochemistry of the venom, it's very, very similar. There's just a lot less research done on it, and because of their ecological niche, people come across them less often. Loxosceles have a nasty habit of hiding in clothing, which means that people come across them a lot more often, whereas Sicarius are not doing that unless you're rolling about in the sand. Desert sand, not beach sand. And so you're not getting the human envenomation events. But the literature on Sicarius venom and its coagulopathy, so how it affects the blood clotting, is very similar to Loxosceles. So we treated them the same way.
Cool. So it's kind of why so many the sellers at some in the UK kind of say it's over ratings only, and they often don't take them to the various shows and stuff.
Yeah.
They're kind of careful with it just in case. Johnson has heard that the male Sydney funnel web is more venomous than the female. Is this true? And if so, is this the case in other inverts?
That's a great question, and yes, this is true with Sydney funnel webs, and it appears to have evolved in this instance, possibly because the males are out wandering looking for females and more likely to come across predators. The weird thing is that it's very primate specific, and there wasn't primates on Australia for much of its evolution. So it's a real quirk, an interesting thing. But yes, it is definitely true that the males are worse than the females. And brings me on to another funny story that the university, I think it was Queensland, were looking for they put a call out for people to bring in male funnel webs to collect venom as part of their anti-venom production. And this particular year, which I guess may be 2018-ish, and we sort of it was in the BBC worldwide, and we thought of sort of non-news. They do that every year. We didn't really think about it, and then a couple of months later, there was a news story that followed on that they'd found the biggest male funnel web that had been found. I can't remember the measurements, but being true Aussies, they called it Big Boy. And I got a call from the BBC, being one of the few venom researchers in the UK, of, do you know much about this story? Can you come and talk about it? So yeah, sure. We don't do that work, but I know what they're doing. And so I went off to the Kent BBC studios and was talking about how they collected the venom and how they're producing anti-venom for therapeutic indications from these spider bites and stuff. Now, when I got back to the office, our company secretary had a wicked grin on her face, and she showed me a screenshot of the live feed that I've just done. And there's a picture of my face, and underneath it said "Big Boy had been found." Which I was glad I hadn't seen the live feed, as I may have corpsed. But yeah, so somewhere I have that screenshot that she sent me, which was quite hilarious. But whether it's the case in other inverts, it's more common, I think, in spiders to be the other way around. Less so with toxicity, but definitely with yield. In theraphosid spiders, adult females produce a lot more venom than adult males do. And their venom starts to run down as soon as they've gone into that maturing molt. And if you look at things like the Nephila we spoke about earlier, the diminutive males just can't carry the venom of the females.
I mean, sometimes, like, how is this the same species? They're so small compared to the females.
Yeah, what's interesting, if you keep them in a large enough collection, you sometimes get giant males. Yeah, so they spontaneously turn up. I don't think they live long, but they so there's a constant selection pressure of keeping the males small.
Harry's found hadagene troglodytes for sale in the UK. They're awesome hadagenes.
Yes, got one upstairs.
They're just, the flat rods they're just a ruin my scorpion.
So
they're really cool. Michael's asked who worked with complex regional pain syndrome.
Yeah, so back in my Pfizer days, we did a lot of work on those sort of complex pain syndromes and things like fibromyalgia, and that are refractive to current analgesics. That's ultimately what we're trying to do, develop new painkillers. It's not something we've done since, but the mechanisms involved are potentially similar to other pain syndromes where venoms are being researched. But yeah, it's not something we're doing directly, I'm afraid.
That's cool. Mate Scott, cheers for joining.
Thanks very much.
Hey Greg, cheers for joining as well.
No worries. Cheers.
So yeah, so as well, then we mentioned right at the beginning as well that you do keep a few yourself for just as pets as well. So you are hobbyists as well as a researcher. What sort of things do you keep as pets?
So on the venomous side, we've got a Lassidoria klugi who is 21-ish years old, I think, something in that sort of region. And we've got Brachypelma and a nice little avic, a couple of royal pythons, a couple of spotted pythons, and some geckos and some more standard stuff. Mr. Pointy is our red belly piranha.
Still going.
yeah, he's a big lad because we moved house recently, and I had to catch them up to move the tank, and I don't recommend moving fish tanks. It's not fun. Particularly not when they're over 300 litres. Because
you don't 400-litre one to move not too long ago, and that was a pain.
Yeah, it's not a fun thing. But because you don't have your fish out as pets, you don't handle them for very obvious reasons. I sort of not realised how big he was, and used the colander as the trusty piranha moving device, because they just shred nets. It's just not worth the effort. And as I offered it up to the colander, I realised that he was about the diameter of the colander. I was like, oh, you've grown quite a lot. So I sort of pinned him against the glass and then went, I'm not sure how this is going to go. So as I turned the colander, he flipped up onto the top, and I just had to bare hand just grab him and stick him in the bucket of water. We're still friends. It all went fine, but there was a moment of a. I don't know what else to do. I've got nothing now. I just had to just grab him.
I can't imagine trying to grab one of my piranhas.
When he's sort of flapping about on the glass, I didn't really have many other choice. Grabbing them from the water would do a whole different matter entirely. But
definitely. So the ones that within the facility of Venomtech, are you keeping them much like people would, just within the hobby kind of thing, sort of very similar sort of setups, or are they kind of very much more basic?
So they're more basic for a couple of reasons, and the main one is that we need to be able to get access to them to collect venom without being too disruptive. So, for instance, instead of having deep substrate for our burrowers, we have a guttering half pipe. So they have a dark burrow, but it's a burrow we can lift the lid on and get them out. With the minimum of disruption, because we can then put the lid back and pop the spider back in afterwards. And we actually won awards quite early on showing that spiders respond to environmental enrichment much like mammals do, with our arboreal spiders, so Pseudomopius and Poecilotheria at the time, and so they need a 3D structure. So we use plastic florist leaves, again a half pipe of guttering, but vertically this time, and vermiculite substrate on the bottom, because it's pretty inert. It doesn't grow stuff. You can see when it darkens and needs changing, and allows us to keep them in a low parasite nature, so we don't have problems with forage flies and those sorts of things. Just keeping on vermiculite with very straightforward husbandry, because we've got a big collection, but there are no doubt people viewing have got similar sized collections. We just use ours slightly differently, and so they made a few modifications, but monitoring their husbandry and welfare is a key thing, because we found, not unexpectedly quite early on, that if you get the husbandry right, you get good venom yields from them. And so we found even with the sort of deep burrowers like what used to be crochet eyes, I can't remember what they're called now, Pelinobuus, that having a half pipe guttering and vermiculite substrate is a good house for them. So
I'd like trying to join it. And there's some, even within the hobbies, but sort of discuss gets us just a bit sometimes, like with your more sort of fossorial ones, substrate depth from that. Actually, we don't necessarily need, 2 foot, 3 foot, whatever foot depths of substrate, just because they'll dig that in the wild. Don't necessarily need to do that in captivity, because you can manage those humidity and temperature gradients, much better. Kind of thing that, they're comfortable. That's the main thing.
It's about getting the microhabitat right, more so than
that's the only reason that something like some of the chelobrachys where are going down so deep in the wild is to get to that particular sort of humidity and temperature sort of thing where they feel comfortable, but you can do that with a few inches kind of thing, or or even less sometimes.
Yeah,
But,
and
hey, Jack.
we feel it's just a lot more stressful to be sort of digging them out from deep burrows, as well as more challenging from the south. So yeah, sort of a basic setup, but it's worked really well for us. We keep monitoring it, and we do a lot of research on how the animals are behaving to make sure we're doing the right things.
That's great. I mean, actually, I'm going to. I sort done sort of some stuff with this, but it's UV lighting with arachnids and
I've
stuff.
done a little, yes.
So, um, it comes up occasionally, not so much necessarily within sort of the hobby, but certainly kind of zoo side of things, which I'm in and stuff. We kind of, gets mentioned every now and then, and some hobbies kind of mention about perhaps, giving UV lighting and stuff. And often we kind of think, oh, the spiders, they're nocturnal, you know, or even sometimes I go, oh, they're photophobic and stuff. But yeah, what's sort of some of the things that have been found with UV lighting?
Yeah, so what we were looking at is to be accurate with what we did was full spectrum lighting. That included the UV end of the spectrum, and working with the amazing Francis Baines, who's fantastic, looking at the UV spectrum, and actually found that it was mostly transmitted through really useful box plastic, which was great for our study setup. And we, again, working with Francis to actually understand the light intensity. So we gave them a lighting that was suitable for dappled sunlight in a tropical forest where the spiders came from. We worked with Hysterocrates in this particular instance, and Heterometrus. And yeah, measured them, their behavior, and also cortisol levels, which hadn't been done before, which is a known stress hormone in vertebrates, but it's also a stress hormone in mussels, the bivalve mollusks. So it's probably fairly well conserved amongst other animals as well, because there's quite a huge evolutionary distance between us and mussels. And then we, I think it was about 6 weeks with the identical setup. And all we did for the second 6 weeks is switch the lights on and carry out the same temperature humidities for the following 6 weeks. And we saw an elevation in cortisol levels. And we also saw changes in their behavior, amount of time they were outside of the burrows, all these sorts of things, suggesting that in those two particular species, they do appear to be photophobic. And although they need a day-night cycle, so they still had ambient daylight from the window, but was not direct in any form, then that light that they're exposed to in the wild is another reason why they burrow deep.
Yep.
Whereas Nephilim definitely need bright light. They like to sit out in the center of their webs. So it really depends on which species of arachnids you're looking at. And I would say that with the peacock jumping spiders, and all sorts of jumping spiders, actually just the native zebra jumping spiders, most of the time I've seen them, they've been out in bright sunlight.
Yeah, and it's something like with the jumpers tend to recommend. Most people tend to go, make sure you keep them somewhere bright, give them a light over the top if they're not in somewhere bright condition. Because I say with the zebra jumpers, you only tend to see them out on really bright sunny days, and then they come out sort of thing all over the walls and stuff. And it sort of makes sense, and it makes sense with things like Heterometrus and or your burrows that if they're burrowing deep in the day, they're probably not. They're probably doing that to perhaps escape light as well.
Yeah, exactly. Yeah.
So, but it's just an interesting thing because obviously, again, not so much in the private sector. And a lot of people will use low-level LED lights or whatever, especially like sort of coloured ones. Often, sort of thing. Even I do and stuff. But in the zoo setting, we tend to have. We often tend to just put UV lights over everything, kind of thing, and stuff. And it's always an interesting kind of topic on that one.
Yeah, I went to they called itself a tarantula zoo in Prague, and this was probably, well, it might be 20 years ago. It was certainly a while ago, and yeah, they had full spectrum fluorescent lights over all the arachnids, but they were out from what I remember. And that the sort of it really sort of struck me. So that's quite interesting, and it's sort of one of the things that started to set the seed of needing to study this. And then we got a great opportunity with Canterbury Christchurch University to actually put a student research project on it and try and answer the question. And I'd like to repeat the study with just using the blacklight UV, particularly with scorpions.
Yeah, because something that again, some zoos do research. We don't anymore, but we used to have a button where public could press the shine a black light so you could see scorpions fluorescing. We don't have that now, but we do have actually one of our visitor engagement volunteers. She actually bought a little black light flashlight. So she's going around, she can just show people, oh, you want to see a scorpion? There it is, sort of thing. That show down sort of in the burrow and stuff. And there was always that thing because, when we changed our buildings and so I stopped the whole button thing because I was like, well, I don't know if it stresses them out or not if someone's constantly pressing the button. And when kids press a button, they like to bang a button as well. So it's like the vibrations of that as well, probably not, that more so that, but there's also that constantly going off throughout the day, constant black light every now and then. So like that, obviously I don't know whether this does anything or not. So
we just don't know enough. That's the thing.
And so, but certainly something that's that's really, really sort of interesting to look at. I know it's something that was sort of brought up within sort of the zoo sector. A while ago, sort of, who does actually need it? We need to perhaps, can someone look at this sort of thing?
They're fairly cheap. The cortisol assay is a little bit expensive, but the rest of the study is quite cheap to do. It just takes a lot of time because you just need to spend time observing the animals in the two conditions, and then trying to make sense of what you see.
That's brilliant. So Jack, I'm guessing, Jack, you're perhaps in the US because you're asking about whether we can keep highly venomous scorpions in the UK. I see Steve's answer with that we need a DWA license, or at least for private keepers we do. I'm guessing actually, do you, for research purposes for your city, is that under DWA as well?
Yes, it was.
And so I know obviously it's sort of different for zoos and stuff, but Dangerous Wild Animals Act license, which kind of varies on council to council how that goes, sort of thing.
That's a whole different topic.
And what they recommend for some people, like since some real thing like what sort of thing.
I've been asked if we were taking them out for exercise in we won't go too far down that road, but I've been asked if I was.
Sorry, Jack is from the Netherlands. Sorry, Jack.
Okay, I've been asked if I was giving my spiders and scorpions daily exercise.
That's it. Oh, we've got another Steve in the chat. Hey, Steve Spiders. It's from the Netherlands, and they don't need a license.
It's UK legislation,
Yeah, all depends on. Some countries you sometimes get jealous of some of the countries that don't need a license to keep those scorpions and stuff. Because some of those are really, really pretty. Some
of the parabuthas, particularly like Transvalicus and stuff, just got amazingly fat epistosamas. They're just amazing. Sort of like topple over.
Yeah, no, I was in. Since really nice. I remember years ago I got called out with someone who village not far from us. They come back from Cyprus, and 2 weeks later found a scorpion running around their bedroom.
Oh well.
Brought it up, took it to the school that they work at, and called out. And I was like, okay, I'll come out and get it. You kind of like, okay, this got fairly thin pincers on it. What's this? So we think we got it down to potentially Misobuthus.
Okay,
sort of thing. It was like, I was like, I'm not sure on this one what this one is, but you probably don't want to be stung by it. Probably shouldn't have brought it to the school.
No.
Just in case. It was like, but came in, stowed away in their luggage, and it was sort of like, well, quite lucky. He's like, obviously, run around their bedroom, walk around barefoot in your bedroom. Quite a lot. And stuff, and how they missed, managed to not get that. So, now for us, I mean, we have nothing that's highly venomous for RD I think it's like tigers, which do license covers.
Yeah.
Sort of thing. So we're our work is a zoo, so that sort of covers most of that. But we have nothing that's like your hots, anything. We don't have any highly venomous snakes or scorpions or anything like that here.
Yeah, but a tiger takes a lot anyway.
Tigers are fantastic. They're dangerous. Well, one of them is dangerous enough. So one probably less dangerous than our wildcats.
Yeah.
Ways, but we're going to test it out.
Yeah.
But I think it's already 10 o'clock, so we've been off for 2 hours. And I don't want to take up too much of your time, Steve. I think if no one has got any questions, we'll probably start wrapping this down. But it's been absolutely fantastic to have you on again.
Yeah, it's been great to catch up, mate. It's been good to chat about my favourite topic.
Hopefully I think everyone has been fascinated to hear some of the stuff that you do, and so on. And there's another thing when people sometimes ask, well, what good are spiders or scorpions and this and the others about that? But there's a lot of medical research that we can gain from these. It's something that I use in one of my daily talks. Is that actually we owe a huge thanks to all venomous animals, because of what good is it? Because everyone only just thinks of they either kill you or it's anti-venom. I don't think of all the other uses that we can gain from it and stuff, even the ones that like I said, are not classed as medically significant.
No, exactly. There's a whole world out there, and it is part of the wonder of the biology and the natural world which we're part of. So it's fascinating. I'm not one of these people who sort of looks at them and go, "What's the purpose of that?" It's like, well, evolution doesn't have a purpose. Everything finds a niche. So and it's great to champion the underdogs of the animal kingdom.
That definitely is, because too many of them get a bad reputation that they don't deserve, and
exactly.
Something I normally say is that actually normally the opposite is often true. What you said here. So quickly on that one actually is the perhaps just think about it, the infamous in the UK, the infamous false widow.
Yes.
And stuff. There's still something that's infamous to this day, of them being the most dangerous spider in the UK and this, that, and the other. But what is their venom actually like?
So their venom is actually is neurotoxin. So they are related to the widow spiders, or Theridae, which are comb-footed spiders. I think is their common name translation of the family. But in the grasslands, there are white widows. We've had them for they've been native for UK for much longer than the false widows. I think that there's a little sideline. They're pretty awesome, because if you look under a microscope, you can see their heart beating because the cuticle's white. And it's like diversion. But the Statehoda, because they are recent in terms of evolutionary terms, they've been here a long time, but it's relatively recent. And yes, we used to say that they're sort of harmless, or when you got the sort of Daily Mail spider that they needed liters of antibiotics to defeat the venom. It's like, if you're using antibiotics, it's not a venom, it's an infection. And that infection may have come from the venom gland, of course, as we now know. But the actual venom itself is not what's causing that much of a problem. There are a few, and they're very, very rare. JP Dunbar and the crew at Galway University are championing this research, and I highly recommend looking into that. So JP's finished his PhD now, but was working on false widow spider venom. So it does cause some minor effects, like headaches, nausea, a bit of reddening, but it's nothing too bad. But what is hitting the headlines is those infections.
Ooh.
So it's key to get good wound care if you do get bitten by one of these spiders, and to not do anything sort of different. If you look at how the Australian situation was, when I first went out to Australia, I was really, the first time I was actually sort of a bit wary of going to a new country, because prior to starting Venomtech, whenever I went to a new country, I'd be always going, sort of diving in and looking at all the local wildlife. But having spent 6 months producing safe working practices, particularly with redbacks and latrodectids, of separation and all this sort of thing, and then to find them on pretty much every picnic bench was a very different situation. And the Aussies deal with it by not being in the way of the spiders most of the time, and ultimately, a few people do get bitten occasionally over there, and there's good anti-venom treatment for redbacks, which are a whole world different to the false widows that we have. But yeah, just let them do their thing, and don't worry about it.
I mean, as well as point, actually, he's held false widows and never had any problems. They're not as aggressive as me. Just they're really not. They're
not aggressive spiders. No, certainly not. But yeah, if you corner them, or they're stick your hand in their web and press them against the side, then they're going to fight back. Same as any
defend themselves, and as anything would.
Yeah, exactly. Yeah.
We've got one last question from Alice. Samopius genus of no exciting hairs, yet are new worlds. Does their venom make up for that compared to, say, Grammosoda or Brachypelma?
I would reckon so. And what you see is that there are very few urticating hairs in arboreal spiders, and I'm wondering, because everything's sort of blowing around more unstable environment, they're probably less able to lift feet up to kick urticating hairs anyway. And evolution doesn't waste, it doesn't duplicate. So you see that greatly in scorpions. Those with big, powerful pincers have much weaker venoms than those with really narrow, skinny pincers. So yeah, when you look at the arboreals, such as Psalmopius and Poecilotheria, then their venom is plenty defense. They don't need the urticating hairs. And then Avicularia, on the other hand, they can actually rub in urticating hairs that they don't brush off. They're just attached and then detached.
With the carabiners as well, like the carabiner versatile as well, sort of thing. They don't kick them; they rub their pipsane onto you.
Yeah, exactly. So yeah, there's quite often one or the other defense is sufficient, and I think that's probably the case.
So, and then with the pokies, obviously it's often cited out like the pokies are the most venomous tarantulas, especially like Poecilotheria metallica and stuff. And I remember either hearing or reading somewhere, I can't remember if it was from you or maybe someone else, that said that there's, or maybe an Andrew Swift, maybe, of potentially looking into whether pokey venom is a bit more similar to things like snake venom, because apparently when it's milked, it's a completely different color to other spider venoms.
Yes, possibly it was me. So yeah, pokey venom is yellow, like snake venom.
Yeah.
And that's possibly a shared enzyme, which we've not discovered yet. And yeah, the term most venomous is also another challenging one, because it for me, my immediate question is, what do you mean? Is it kills the most people? Is it the most toxic? Or is it has the most venom? And for me, most venomous really encompasses has the most venom. It is my sort of linguistically looking at it. People are generally want to know whether it kills more people or most dangerous. But pokies, I would say, are the most venomous tarantulas we work with in terms of having the most venom per body size. They definitely have a lot more than a comparable size Psalmopius, for instance.
That's brilliant. Cool. Okay, well, yeah. Before we go down another sort of stuff, we could just chat for many more hours, I'm sure.
Yes,
but
indeed.
Conscious of time. So yeah, a massive thanks again for coming on again, Steve. I'm sure hopefully again sometime in the future I'll have to have you back on again.
Yeah, no worries. And yeah, it's always great to catch up, and it's it's a favourite topic of mine. And
something that especially like in the hobby side of things, often people don't hear about this sort of stuff.
Yeah,
Exactly. Especially about sort of the animals that they're keeping and stuff like that. The animals that they keep are used in medical research and stuff, and various properties of their animals, their pets and stuff is like super fascinating and stuff. So yeah, massive thank you. And thanks to everyone that's in chat. Thanks to again, another thank you to Lewis and Eddie for becoming IcePod members. So and hopefully we'll see you all again next week. Got a video coming out on Wednesday, which is should be a shop walkthrough of walking the wild side exotics or walking the wild side reptiles and aquatics and native buzzard. And so me and Steve, though, we popped down there yesterday. So to have a quick look. And I believe Scott is live tomorrow with Tim Baxter talking all things scorpion and stuff. So that'll be really interesting. I think Tim's got something like 50 species of scorpion.
Oh, fantastic.
That he's hoping to breed.
Yeah,
excellent. It'll be fantastic. Oh, so yes, Steve, I am vending at Seas. I'll be at Seas. Will you be at Seas, Steve?
Yes, walking. Not vending, but I'll definitely be there. So yes,
see you there. Yeah, I'll be there. What I'm taking, not entirely sure, probably metallic emerald cockroaches because they breed like the clappers for me. So probably be taking some of those. And I'm not sure what else yet, sort of thing, but I'll be there. So yeah, coming up and down and stuff, and I'll have a few bits, I'm sure. Guys, until next time, cheers for
I'll see you there.
For joining, and I'll say good night.
That's on. Cheers, mate. Thanks very much.
Everyone.